Our first lesson this week is on microscopes, where we've moved into Unit 4, which is all about the cell. So we'll be talking about how the cell works, the different parts of the cell are, and then we will get into the specifics about two different processes, photosynthesis and respiration, which are how the cell breaks down energy and then uses it for other things, or makes those energy molecules if the cell is part of a plant. So this first assignment is called Microscope Parts. And it says before we can begin the study of cells, it's important to understand the equipment used to view a cell, which is a microscope. So in this assignment, you're asked to view this movie, which goes through the PowerPoint presentation, and then focus on these questions. Some of them are answered in the presentation, and some of them you have to use some common sense for. For example, number 1, what type of microscope is most often found in schools? For example, what would you use in a science classroom in the high school? What do the lenses of a light microscope do? How is a stereoscope different than a regular light microscope? What does a SEM use to produce more detail of the organism? How is a SEM different from a TEM? What's the major disadvantage to using a SEM or a TEM? It's the same disadvantage for both. Why would someone use different types of illumination on a microscope? And then I'm asking you to simply label the parts of the microscope pictured here. So let's go ahead and take a look at the microscope and its parts. There are several types of microscopes. The first is called a light microscope. This is the model found in most schools, and it uses compound lenses to magnify objects. The lenses either bend or refract light to make the object beneath them appear closer. So the lenses are found in this area and as well as here. The object you're viewing goes here, and as the light, which comes from here, shines up through and then through the lenses to your eye, the lenses bend the light, which magnifies the object. The common magnifications found on a light microscope are 40, 100, and 400 powers. But you can add a drop of oil to the 400 power objective, which can improve the quality of focus and magnification at that highest magnification. A stereoscope, as you can see, looks a little bit different. This allows for binocular viewing of larger specimens, which basically means you get to use 2 eyes instead of 1. It usually only magnifies 10 or 20 times. It can be used for thicker specimens. You don't have to, if the specimen you're looking at is thick, for example, you're looking at a whole insect, an entire insect, you don't have to cut anything. And it creates a 3D view of a specimen. It's basically a bigger, higher magnification, easier to use magnifying glass. The next microscope is called the Scanning Electron Microscope, or SEM. This allows scientists to view a universe that's too small to be seen with the light microscope. So this isn't available in high schools. They're large. They cost thousands and thousands of dollars. They don't use light waves. Instead, they use electrons, which are negatively charged electrical particles. We talked about electrons when we talked about atoms and elements in our last unit. And they use these electrons to magnify objects up to 2 million times. The SEM also creates a 3D view of the specimen, but the disadvantage is that you can't view living things. In a light microscope, you can take a drop of pond water and put it under the microscope, and you can see the organisms from the water as they're still living. You can see them moving, swimming, things like that. Here, you cannot view living things. The process to create the specimen would kill them. But these are some scanning electron microscope images. Usually this is attached, the microscope's attached to a computer, and so you don't look through an eyepiece, you actually view the object that you're looking at on a computer screen. And then of course you can take photos of that and print them out. This electron microscope is a little bit different. It's called a transmission electron microscope, or TEM. It also uses electrons, but instead of scanning the surface, as the SEM does, the electrons are passed through very thin specimens. So to remember TEM, think thin. In the TEM of a cell, you'll notice that you see the inside of the cell, not the surface. So you actually have to cut the specimen into extremely thin slices in order to use it with a transmission electron microscope. There are many types of illumination in microscopes. This is talking about how light passes through the specimen, which can change the view of the specimen, making some parts easier to see and others not as easy. So you can see if you change the amount of light or the type of light going through the specimen, it changes how the specimen looks. All of these are microscope images of, it appears to be cheek cells. And you can see if you change the light, it makes the cheek cell appear different, and then you're able to see different parts of it. So let's review the microscope parts. This is called the body tube. This is the revolving nose piece. It's called revolving because this part here will turn, allowing you to use the different, next word, objective lenses. These lenses hold the different power lenses. Here are the stage clips. These hold the slide in place. This on the bottom is called the diaphragm. It's used to change the amount of light that goes through from the bottom, which is the next one, the light source, up through the stage and then through the microscope slide, through the lenses and up to your eye. The part where you look is called the eyepiece. It's also called the ocular. This is the arm, which supports the eyepiece and the body tube and the nose piece. The stage, which is where you set the specimen, and in order to focus it, the stage actually moves up and down. There are two ways to focus, either the coarse focus, which you would do when you first start looking at the specimen. You would get it into focus, for the most part, and then once it's focused with the coarse focus knob, you would use the fine focus to get an even clearer picture of each part of what you're looking at. And last but not least, the base, which is what it sits on. Magnification. Your microscope that you would use in high school has 3 magnifications: scanning, low, and high. Each objective will have the magnification written on it. And in addition to this, the ocular or the eyepiece also has a lens in it, so also provides a small amount of magnification. To get the total magnification, you simply multiply the magnification of the ocular, which is 10, times the objective that you're using. So the scanning objective, or sometimes called the low power objective, is where you're just basically searching for the part of the specimen that you would like to see. It's going to be your smallest magnification. Then you switch to the next power, and you can get an even more detailed picture, and then lastly to the high power, which is also sometimes the oil immersion lens, depending on what is attached to the microscope. There are a variety of resources for you to view. If you choose to click on any of these, I can make the PowerPoint presentation available, and you can, actually I'll make the links available in the assignment if you want to go through and take a look at other microscopes like phase contrast microscopes, fluorescence, SEMs, TEMs. You can visit photos that have been taken through these microscopes of various things at any of these resources. So that's Microscope Parts. If you have any questions, as always, send me a message or an email.